different types of animals at different depth bands. And so we've got a fairly good picture of some of the species we find at about 500 meters and above, which is about where we are now descending. But we can get into much more kind of exciting, interesting coral species like the bamboo corals and the black corals, and of course super lucky, a group called the chrysogorgid corals, which are very beautiful at these much deeper depths. And we will also see different fish assemblages than we would shallower. But this depth will be past the max depth of our West Coast Trawl Survey, so we could see some interesting things out past that, and some things that aren't usually seen along the West Coast, so that could be exciting. So the Trawl Survey is a program that NOAA uses to help monitor groundfish fisheries. And as a coral biologist, it's one of the ways that I get a lot of my samples as bycatch, but it's always a pleasure to be able to work with ROV teams like the Nautilus to actually see what these habitats look like in real life, in person, and what the animals look like on the seafloor. Before they spend time in a trawl. Yes. Although some of them come up looking just basically like they do on the seafloor, but not all of them. But we also get the context of the type of habitat that they're living in. We can measure things like how high the rock structures are, what types of currents there are. If the habitat is what we call high relief, which would be really steep and tall, or low relief, just like flatter but still hard bottom habitat. So it's all context we get from the video feed that we don't get when we're just looking at the animal either in a museum or in the lab. Some interesting fish swimming in the background as we descend.
There's a big siphonophore, a couple of them actually.
Mm-hmm. We also have a team of scientists ashore who are joining us. And so we have we have a science chat where other scientists, some of whom will be on watch later tonight through this dive, and others who are just joining to help with collections and identifications are communicating with us and communicating with other scientists on the ship. So we'll be able to get identifications from some of them. Some of them will help us identify things we want to collect through the dive. So welcome to all of the scientists ashore.
And thank you for being here.
Yes.
What a way to spend a Friday night.
Mm-hmm. The Nautilus has been spending a lot of time in the National Marine Sanctuaries as it travels down the coast. They did start up in Canada working with Ocean Networks Canada, but then they visited Olympic Coast National Marine Sanctuary, where they did deep dives in Wanda-Fuca Canyon. They then moved south, and they've been doing work in Greater Farallones National Marine Sanctuary and Monterey Bay National Marine Sanctuary. We've done a couple dives in Channel Islands already, and now we're moving up to, we're out on Santa Lucia Bank, which is not currently a sanctuary, but is part of the proposed Chumash Heritage Marine Reserve. And so some of what we'll be doing tonight is just establishing baseline data for what's here as part of the process of possibly setting up a new marine reserve here.
So what characteristics does an area have to be considered, or to qualify, I guess, to become a marine reserve?
Can you repeat the question? I just dropped off of V-Link for just a second.
I was just curious for, I don't know how official it has to be for an area to be considered a marine reserve. Like, are there certain qualifying characteristics that you'd be looking for to make it a reserve?
There usually are. So usually a group from the public will propose a new area, and then it has to go through a review process that involves things like these dives where they establish kind of the uniqueness and the characteristics of the area, and then it goes through several layers of federal review. And Lisa is reminding me that the sanctuary is not a marine reserve, so it's separate from the marine reserve system. So this is a new proposed marine sanctuary.
Cool. And sanctuaries and reserves are different things, correct?
Yes, they are different things.
Right.
So Lisa says most people consider a marine reserve as a no-take fishing area, and for a lot of the marine sanctuaries, fishing is actually one of the allowed activities.
Ah, interesting.
But different sanctuaries do have different rules as far as what types of fishing and when they're allowed.
Yep.
And sanctuaries can also contain reserve areas.
The individual fishing regulations are designated and implemented by fishery managers, such as the National Marine Fisheries Service and the Marine Fisheries Councils. And it looks like someone is actually, you might be able to take some public questions tonight.
So here's a quick question for Marley. Is this video being recorded in ProRes codec as well, or just the H.264?
It's being recorded on several different formats. We've got two main recording folders, H.264 and, well, the full 4K. That's what hold on, I'm looking at our folders right now. Yep, we have both. We have an enormous amount of files. Is what it what it equates to.
Thank you. That was from one of our
of data.
scientists ashore.
Science, do you guys want to tell us a little bit about your backgrounds, about how you got into doing this type of work, or how long you've been doing what you're doing?
Sure. So as I said when I introduced myself, I'm a geneticist at the Northwest Fisheries Science Center. I actually came into genetics by way of marine biology. I had known all through high school that I wanted to do marine science. And as an undergraduate, I got my degree in oceanography from the University of Washington and had the opportunity to work as an undergraduate in a molecular biology lab run by the oceanography program, working on the genetics of phytoplankton, and got really interested then in using genetic tools to study how marine animals interact with their environment. And so just have continued on that path ever since. And the nice thing about working in genetics is you use the same tools to work on everything from bacteria to primates. And so I've had the opportunity to work on everything from phytoplankton species to corals to fish. I've worked on salmon and even done a little work occasionally with the marine mammal program. In the last 8 years, I've really dialed in on coral because I find it a really just fascinating system to work in. And working in these deep sea habitats, it's like ground up science. You're looking at species that are new and that are very poorly understood and just starting to understand their really basic biology.
Yeah, there's still so much to be discovered. That's awesome.
We're so happy to have Meredith here to puzzle some of this out and tell us what we've been seeing. It's been really great to have her on board. This is Erica. I've been working for NOAA Fisheries for the last 18 years. I moved to the West Coast to go to graduate school at Oregon State University and got a master's degree in marine resource management, which led to a job as a fisheries observer. And that led to the realization that I loved going to sea and actually seeing something new and different every day. You never know what you're going to find out there, and you're going to learn something new every day. The observer job turned into a job with the research team at Oregon Department of Fish and Wildlife. And that turned into a job on the West Coast Trawl Survey that we talked a little bit about earlier. So I did that for 10 years. The Northwest Center charters commercial fishing boats to help us sample from the Canadian border to the Mexican border 2 times every summer. And those numbers are used to create stock assessments and tell us what the fish populations are off the West Coast. And in 2011, the Northwest Center developed an autonomous underwater vehicle with the Pacific Islands Fisheries Science Center. And we've been able to take our little robot pretty much all over the world. I've been able to go to Guam and American Samoa with it. We've done work in the Gulf of Mexico. We've done work up off of Vancouver Island as far out as 200 miles offshore at Cobb Seamount. It's been a great project and has enabled me to see lots of new species in new areas.
How many times have you gone out to sea? Do you know?
I have over 1,000 days at sea.
Yes, epic. I love it. That's great.
And Erica is one of the folks who any of the invertebrate fish, anything that you're looking at, generally can tell you what it is from all the work she's done as an observer. She has extensive experience looking at all of these animals. One of our scientists ashore is helping us out with the public questions tonight, and so she's opened apparently the questions and will be kind of prompting us through the dive as time allows. I said that the questions are already coming in.
This is a radio check. from that person. This is Claire. Can y'all hear me okay?
Hi, Claire. Yep.
Hi, Claire.
Yeah, we hear you loud and clear.
Hi, friends. Beautiful. Well, we do have some questions. One of them is, are we going to go beyond 1,000 meters today?
Yep. And we've actually already passed 1,000 meters right now. We're at 1075. Tonight's dive will go all the way.
Our target's what, 1,500 meters?
1,500 meters. Yes.
Copy that. Another question came in 2 minutes ago, and this person is wondering if we're planning on collecting any samples during this dive.
We hope to. We have a master collections list. We have some target coral and sponge species to collect, and we'll also be hoping to collect some sediment cores right after we land.
Perfect. During this time, since we're just diving, we do have a question for the pilots that maybe someone can address. It says, what is a topside reset, and what technology is involved during that process?
Sure. Yeah, we can go ahead and take that question. Topside reset. Topside is a way that we refer to kind of the user interface with the ROVs. Includes GUIs and things like this. And the thing about that is that if we refer to a topside reset, we might have to relaunch, reopen these kind of .exe files or rehit those in order to have our GUIs spin back up and be responsive to us. That's all that means when we say we might have to reset topside. Yeah.
Thank you. It sounds very official, so thanks for explaining that.
Sure.
Only 400 meters to go.
Roger.
One of our scientists ashore is asking if we can get a Zoom on a full missus. Unfortunately, while we're on the descent, unless it joins us, I'm not sure we'll be able to get a Zoom, although we do occasionally see them while we're on the bottom and might try to on them. Full missus are the white. So we'll be keeping an eye out.
Science, you just, not sure if it was Meredith or Erica, but one of you just broke up a bit. Can you repeat what you just said? Sounds like
Oh, it was Meredith. one of our scientists ashore is looking out for solmices for a zoom if one happens to float by when we're on the bottom, to get a nice zoom on it. They're the jellyfish-like creatures out of the white discs with many, many arms around.
Cool. We
Yeah,
will
those
keep
are
our eyes peeled.
Did you say Solmaris?
Okay.
Or what was the jelly name?
Solmices.
Solmaris. Is that it?
Solmissus.
Oh,
Solmissus.
Solmiss.
I'm going to
With
break
the
the
caveat
silence.
that I may,
Oh, sorry. Go for it.
I was gonna say, with the caveat that I may be butchering that Latin.
Sounds like how I pronounce that.
I think it sounds very official. So we do have a question about the octopus gardens. Apparently there was a sampling crate there, and someone is asking if it has been retrieved.
Can you say again what was if what has been retrieved?
Yeah, the question is, are you going back to the octopus gardens to retrieve the sampling crate? I think that so far information I'm getting is that's a negative currently.
Yeah, I don't think any of us really have a good answer for that up here. So it might be more of a question for expedition leaders and things like this, that one.
Yeah, I think answers are coming in on my end. It says not on this side. So whoever asked that question, the answer is not on this side.
Yep, we are much further south than that here right now. That's up in Monterey Bay National Marine Sanctuary, and we are down just north of Channel Islands National Marine Sanctuary. San Lucia Bank is just north of Point Conception.
We have a friend online celebrating that Argus got repaired, but they are wondering what had to be done to get Argus back and operational.
A whole lot. Okay, that's a bit of an overstatement. We worked on, we have different things we call bottles, and we basically worked on repairing some parts within the bottles and put it back together. So
It sounds fun when you say it fast.
Yeah, you make it sound like it was so easy.
But basically, what Jess refers to as a bottle is like a 1 atmosphere cylindrical housing. And what would you say, about a foot in diameter?
oh, yeah.
Foot, I'm showing my age, right? It's a scientific community. How much is a foot in kilohummels? It's about 3 feet long. It's quite a volume. It's impressively heavy. Basically, we took the insides of it out. The head of the cylinder comes off, and the electronics is packaged in a frame that's attached to the lid. So we took that out and took it in the house. Oh, I left out a part. The housing flooded at depth, and there was still power on it. So seawater in the housing, there's some, I guess, electrolytic processes take place. There's gas generated from the seawater, and all of that at depth. So when we bring the housing back to the surface, it's potentially dangerous. There's a lot of internal pressure. And the housings and their various penetrators that allow electrical and fiber optic signals to go in and out of the housing. Those penetrators and the housing itself are designed to support external pressure, not internal pressure. So it's not really an overstatement to say that it's potentially a bomb sitting on the deck. And Dan Cormany, another ROV tech in characteristic style, walked up to it and stabbed one of the cables with a knife and let the pressure off. All right. There were vent ports on the housing itself, which cracked the vent ports and let the pressure down until it was safe. Opened the housing and dumped out a few gallons of seawater. So that's where it started. Back to, we took the cap of the housing and the frame containing electronics into the shop and spent the next, 40 hours?
Yeah, somewhere around there.
Rinsing, cleaning, separating parts that were recoverable, reusable from parts that were completely destroyed.
Yeah.
We had parts supplied from shore via water taxi. And under Bob's watchful eye, boundless enthusiasm and what's that other word? Optimism?
Optimism, for sure.
Bob never for a minute doubted that we weren't going to get this back together and get it working. He may have been alone in that, but it's, we replaced a lot of parts, rewired it, put it back together, tested it on deck, and it came good.
Yeah, it was a good couple days of work. It was a good couple days of work, that.
And nights.
And nights. Looks like we're about
Having
100 meters altitude off the bottom. So science, if we start talking about ops, we'll have to hold off on some questions until we get ourselves situated on the seafloor.
Having flooded housings before, I just wanted to say congratulations and quite impressive, guys.
Thank you.
Thank you. And you.
And you.
And you.
All right, so I'll stop 050 when we get there. And then we're going to be heading up 075, correct? To our next target?
Well, so we're pretty close to that 4V, so we could just probably go due east and head up slope and then continue along the contour.
Sure thing.
And yeah, we're going to be heading generally towards like 125.
Okay.
So if we land in soft bottom, right after we land, after we're situated on the bottom, would that be a good time to just go ahead and take a sediment core before we start the ship moving towards the first waypoint?
Yeah, sure thing.
Yeah. We'll see what kind of bottom we land on. We're right.
Yeah.
Right on the edge of this.
All right. I'll stop 050. All right. Great. And since I'm pretty close to east, I'll have you turn your heading to face 090. I'll just back down and back up a little bit, and we should be good. Actually,
I'm going to switch you to DBL.
Sure thing. Thanks, Ben.
Yep.
All right. Get all those pyrosomes we had hanging out with
Still
us.
here.
All right, Kim, let me know if you got any spin on you. Oh, you're already there. There we are. Nice.
Still coming around.
Roger that. Spin. All right. We'll go on down here, then acquire the seafloor. Sorry, guys. All right. Coming down. There you go. There's the seafloor now.
Right where we left it.
Right where we left it. Huh.
I always love that first view.
All right, well, we'll get ourselves situated. We'll see if this is coreable. But first, let's do a white balance for Marley there. I'll get a little closer to the seafloor. Maybe I'll just park it. All right. Thanks, Kim. Does that work for you there, Marley?
Yep, that's perfect. Thank you.
Sure thing.
All right, camera is going to go black just for a minute. I'm always nervous it's not going to come back. All right. Perfect, we're good.
Roger that. Science, I'm not sure how horrible this will be. We can give a poke to the ground right now since we have the arm out anyway, but all these rocks, we can try.
Yeah, it doesn't look great for core.
No.
No, it's pretty cobbly.
Yeah, we can kind of.
The good old scientific poke.
Yeah, it's pretty, as you say, cobbly. That's a good word to describe it.
Guy Cochran is online with us in the science chat, and he says 0 chance of a core.
Roger that, guy.
Yeah, I think we anticipate landing in softer sediment. So we'll go ahead and start moving towards the waypoint. And if we find some soft sediment, we'll take one. But otherwise, you can only take one if the habitat is available.
Roger.
All right, so we're going to go upslope 090. I think we're going to go about 120 meters and then more gradually go upslope for a longer distance to the next target.
What's the distance to that target? It's a couple 100 meters away.
120 meters.
120 meters, roger. 090. Well,
090.
bang on that heading anyway. That's great. Go ahead, Ben.
Yep.
And is that first target 4V?
Ben, if you want, since we're so deep, we can do like a 50 meter move for that. Looks like it'll be pretty calm for the first little
Yeah.
bit.
Seeing the Argus meso.
Yeah, great, thank you.
Hoping we'll get some slips though.
Some deep sea holothurians. These are those gray arcs. They're a deep sea cucumber.
Oh, these are cool.
Move it.
Riley, gonna do a quick zoom on this guy?
I was just about to ask if we could.
If you want, I can sit down while we wait.
Yeah.
So we're getting the ship moving and waiting for that move to transmit all the way down the cable to Argus and then Herc.
That is correct. Yeah. Do a quick bump up here.
So one of our scientists ashore gave us some more information about the National Marine Sanctuary that is proposed. And it's
Cool.
stretches from Cambria, south along the San Luis Obispo and Santa Barbara County coastlines to Gaviota Creek, and then seaward to include Rodriguez Seamount, Arguello Canyon,
come wide,
and
please.
Santa Lucia Bank.
Riley, come wide. Thank you.
San
And the area is
Lucia Bank is
located,
where we are now.
and the areas, so this is located between the Monterey Bay National Marine Sanctuary to the north and the Channel Islands National Marine Sanctuary to the south.
It's a different color behind that anemone.
Yeah.
Kind of like a mushroom.
Is it organic?
Looks like it.
And then
I'll get a better landing spot for us and we can see.
wonder if its polyps are retracted or something.
Yeah, it looks like it might be
Disproportionately
the anemone.
Oh, Raj. Riley, you want to do a zoom?
Roger. Cool.
It's a big anemone.
Looks like it over it.
It's a python anemone.
I wonder if it's eating something, digesting something inside.
Maybe.
I think those brittle stars might agree with you.
Thank you.
Would you zoom on the shrimp to the right there? Can we
Oh yeah, sure.
see him from here?
Oh, this one here on the left?
Excuse me, other right, yes, the left.
Happens to me all the time. Roger. All right, Marley, go ahead.
Thank you. Oh, I heard we were coming. Hey, buddy.
Square us up a bit more. Look at those big antenna.
That guy is probably a Pandalopsis ampla. We see them in.
Yeah, very, very bouncy. I think it's a swell. These hues are pretty good. All right, Marley, you want to come wide, please? Thank you.
There's a cute little cup coral right there next to him. Possibly Javonia with the shape.
All right, I'll get out in front of Argus a bit and we can play around some more.
I'm used to landing in the mud when we're off walls, and it's nice to see this cobbly bottom.
Yeah, it means only good things are in store.
Mm-hmm.
And to our friends watching on the other end, we are open for questions, so go ahead and type them if you have any.
Lots of different sea stars down here. I've seen a Dipsacaster and what used to be called Zoroaster. I believe that genus has changed.
Is that the thin white ones or the fatter white ones?
The fatter white ones, I think, are the Dipsacaster, and then the thin ones are the what used to be the Zoroaster.
We just
And
went over a sea spider as well.
Oh, it was a big one.
Are we stable enough to get a quick zoom on that shell hash to the left?
Sure. All right, Marley, go ahead.
I don't actually know that this is hash. This might be very small snails hanging out in the sediment together. All right, Marley, go ahead and come wide, please. One of our scientists ashore, Dubo Lindsay, suspects it may also be a dead and dissociated urchin, possibly not snails, which also makes sense. We've got a grenadier.
That current.
Yeah.
Scarlet king crab, Lithodes scusi.
Those are some impressive spines.
30 works, thank you. like cobbly rock.
That's quite an impressive rock pile. It almost looks like basalt, although I don't think Santa Lucia is volcanic.
We actually had a question about the rocks. The question was essentially, how do all those rocks get that far into the ocean? So even though we're all into the biology, if anyone wants to speak to this geology, we do have a question.
Oh, yeah, Lisa just called on Guy Cochran. I was waiting for him to chime in also.
We do have a geologist amongst our scientists ashore who may be able to fill us in on more of the history of why we have this really impressive rubble field here.
It's Pacific Flatnose just going out to the right.
Nice thing with all of this rubble is that it is definitely hard bottom habitat, and this is the kind of material that you will see the deep sea corals and sponges colonizing. Although we haven't seen any yet so far, I'm seeing tunicates and holothurians, which are sea cucumbers. So Lisa, who was on with us earlier, has chimed in that the Santa Lucia Bank is primarily made up of well-bedded sedimentary rock, which is a result of the cementation of small particles. In the Santa Lucia Bank, these are cherts and siliceous rocks. Additionally, there are small pockets of early tertiary sedimentary rocks that have deformed and eroded and are embedded in the seafloor.
Look at the little snailfish.
Mm-hmm. That's the second one I've seen go by so far. So hopefully one will give us a really good photo bomb. Got looks like a purple holothurian tucked down on the rocks there.
We have 15 left. Great. I think it's all right, Ben. You can do another 30. Oh, I don't see anything too crazy in sonar.
Yeah. All right.
We can always just wait till we get to the top of this guy, and then, but it seems to be kind of diagonal like that.
Yeah. Yeah, it should, the strike should be a little like 08007.
I have many more questions, and in this silence, I'm going to interrupt with one more. It's a fun one. This question is,
Sure.
my daughter asks, what is the typical variety of sea life expected at this depth?
We were seeing some of it. These depths, the echinoderms, which are the sea stars, the sea cucumbers, and the sea urchins are some of the most abundant animals. So that's what we're seeing the most of. We have seen a few deep sea fish go by so far. And hopefully at these depths we will start encountering some really interesting corals and sponges, but they can be more sparse. The sea stars and sea cucumbers actually at these depths are so abundant. They're one of the most common animals on Earth.
Go ahead, Ben. Okay, great.
Those red
You
little
can see.
stars are quick.
Yeah,
Sorry.
they're moving fast.
Yeah, so we're pretty much at the depth contour for 4V, and we can keep on going up slope for a bit. There seems to be a little flatter edge in this feature, maybe 50 meters east of us, but then we can start heading down south to the next target. So
Can you
does that
keep
sound
coming
good?
up there a little, please?
Science.
My V-link dropped for a minute at the beginning of that, so I heard. So can you repeat the first part?
Yeah, so we're pretty much at that target, the launch target 4V now, in terms of our depth, and so we can start heading to 4W. But if you want to keep on going up slope onto this little embankment, if you can see high pack now. Basically, if you want to keep on going east upslope for a bit and then head down to 4W, or just start kind of going more along the contour.
How much more
Yeah, why don't we get up?
depth would we gain?
So the next one's going to be about
here.
Yeah, so the next target's going to be about 40, 50 meters shallower, but we can keep going up slope. Here, there seems to be a little less steep part that we can kind of start moseying down once we get there.
Yeah, that sounds really.
Okay.
That sounds really good. Let's do that.
Okay, great.
Okay. So we'll continue on this until we get to that platform.
Yeah, I'm going to put in another 30 meters.
Sure.
Please. Great, thanks.
Really nice boulder field here. If only it had some corals and sponges.
So when we're analyzing both this video and the still images generated by the AUV, we quantify the habitats that we come across by the size of the different particles. So we have mud, and then the next bigger one is sand, and the next bigger one is, I believe, gravel, and then we go up to pebble,